Hi, everyone. Thanks for coming to Catalyst Episode 2. My name is Greg. With me, we have Kristen. Hi,
Hello.
Kristen.
Hi.
This is a show where we are talking about science news, cool things about science, hopefully informing you about science a little bit more, making science a little bit more approachable, and then debunking science, conspiracy theories about science online. Up this week, we have some news about COVID-19 and the mink population, and we also have some cool fossil evidence about how our ancestors, human ancestors, might have dealt with climate change, and some other cool science news. So, and talking about the vaccine, some news about the Pfizer
Yes.
COVID-19 vaccine. So, um, first up, we have a video on COVID-19 and what's going on with the mink population.
We'll continue to talk about the election, but all of this, of course, Dr. Jen, is happening in the backdrop of a pandemic.
And the
40.6 possible
infectious diseases, this information is just coming in. Viruses infect animal species, including weasels, of which mink is part of.
So yeah, coronavirus is SARS-CoV-2 is a coronavirus that causes COVID-19, and that that is something that happens naturally in the environment. SARS-CoV-2, the coronaviruses are everywhere. They infect pangolins and bats, and that might have been one of the sources for SARS-CoV-2, which is the source that we know that is actually causing COVID-19 in humans right now. And it's just something that, like, it's a difference that's important, right? It's like the reason why we have HIV and also AIDS. The names are different because they're two different things.
Yes.
And we have here we go. So it's interesting that the minks in Utah were being slaughtered after the virus was detected, which kind of means that they had some genetic testing and molecular testing going on with, like, I wonder if like all the farm animals were doing that, or if it's just minks, or I mean it pretty much has to be all farm animals, because we wouldn't know at right now what could be it's called like a reservoir where the virus could actually be for a long period of time and then mutating. And that's the biggest fear, right? It goes to another population, mutates, and then comes back, and now we can't deal with it.
Yeah, it makes a second crossover into humans, and then now we have another pandemic to worry about.
Exactly, yeah, second pandemic with a second mutated strain, exactly.
Family. In September, here in the U.S., mink in Utah were slaughtered after some strains of SARS-CoV-2 were detected in those animals out of an abundance of caution. Now in Denmark, the Prime Minister has just reported to the World Health Organization plans to slaughter millions of mink after a mutated strain of SARS-CoV-2 was detected in mink and then found in at least 12 people in Denmark. So again, out of an abundance of caution, there has not yet been any scientific data released or peer-reviewed yet. So it's difficult to really do a deep dive on this. The World Health Organization is investigating.
Yeah, really interesting. I mean, it just goes to show you the importance of microbiology, molecular and genetic testing right now, especially in the rapidness, like in order to get these tests done, but then also to really go and do the research into new populations that we wouldn't think would necessarily be where we would think. So it says, the New York Times article that they're referencing here, in Denmark, the virus has shifted from humans to mink and back to humans and has mutated in the process, which is what we were talking about, which is very scary.
Yeah.
Because with 12 people so far being found, and had less response in antibodies and lab tests, I mean, that is extremely dangerous.
Yes.
Because that really gives it's really it basically, hopefully, basically right now they are in the stages of this new mutated strain, basically where the world was back in November, December of 2019, where we found that the SARS-CoV-2 existed. We had tested for it, seen the problems, but it hadn't gone to community outbreak yet, although it was kind of like outbreaking in China at the time, obviously, but and then later Europe and then globally, but
Yeah, but it's already a second crossover event,
yeah,
so
exactly.
it's
So it's
and
hard to know.
then
I mean, hopefully, I mean, basically right now, all of Denmark has to, you basically have to test everyone that's and do contact tracing. That's really the only way. Everyone has to get tested for this new virus, and it's probably a different test too, because what they're using is to detect this is probably something called a qPCR test, where you're actually having DNA bind to one another in a test tube, and then seeing if any DNA amplifies that you're looking for, a little flag comes up, the fluorescent marker comes up. So why that's really big is how are they going to detect the SARS-CoV-2 versus, I don't know, I mean, this isn't SARS-CoV-3, I'm not like making any declarations, but let's just call it SARS-CoV-3. How are they going to detect that in the qPCR test? Because you wouldn't want a false positive that thought it was SARS-CoV-3 when it was actually SARS-CoV-2. Does that make sense?
yes, yeah, that makes sense.
So the genetics need to be known and different, because they could have different outcomes, especially if they have a different response to antibodies and stuff.
Right,
So
a lessened response. So
exactly,
no bueno.
exactly. So basically, the difference and how you do that and how you tell the difference is through genetics. So you look at the sequencing of them, you have it come back, and then you see if they align to which one, SARS-CoV-2 or SARS-CoV-3 that we're calling it right now. And that's actually a pretty cheap, pretty easy thing to do. Sequencing is very cheap now, and it really isn't anything. It's no cost compared to the PPE that this person is wearing right now. Let's just say that is more expensive than the $4. Well, it's more than that. Let's say.
Wish we had that in the hospital.
Yeah, that's a good.
Pretty
And that's
good.
like WHO probably.
Yeah.
Looks like it. Oh, it's the Danish veterinary and emergency management officials.
Well, maybe everybody will be wearing mink coats after the pandemic's over.
Yeah, it's definitely a faux pas with the. Yeah.
Yeah. Maybe in this case, no.
So yeah, the chimpanzees have suffered from outbreaks of other coronaviruses. Yeah, I mean, so the reason why like SARS-CoV-2, the hypothesis right now for why it's so incredibly good at binding to like the outbreak is basically we have a type of cell in our lungs that has a little flag on it, and that the SARS-CoV-2 has a really good affinity to that flag. It's called an ACE2 flag, and it's binding to that at a really high affinity, and it's because as we think, because of this elongation that happens in the genetics, we can actually sequence it and compare it to other coronaviruses, and this one specifically has like a really long insert, and the hypothesis right now is that insert is why it's so good at binding to the ACE2 flag. Mm-hmm. But the likelihood of that particular part of the genome mutating is fairly low, right? It's because it accumulates mutations through, it's an RNA virus, but it's going to accumulate mutations randomly across its entire genome. So the likelihood of that specific mutation happening right where that binding is low. So that's why the hypothesis right now is actually that this SARS-CoV-2 would actually mutate slower than the normal coronavirus that we find, and that you have to take a flu shot every year for.
Right. It does mutate just slowly, and we're able to keep up with it, is what I've heard.
Exactly. Well, yeah, in theory, that's what our hypothesis is.
Right.
But yeah, it's good that they're taking swift action. I mean, it's sad to think about all these millions of minks dying, but I mean, there really isn't anything that they can do. But right now, I'm actually more worried about those 12 people that are infected.
Right.
Like, and they have, I mean, not only contact trace, but I mean, they quarantine.
Yeah, they need to be full on isolated.
And wait, you actually have to do more than that with the contact tracing, like more than this entire town. I would do genetic testing for of this SARS-CoV quote unquote 3. Like
Yeah.
So the WHO is not SARS-CoV-2 mink associated variant strain.
Not good. We want to eventually get out of this
Wow.
pandemic.
Since June 2020, 214 human cases of COVID-19 have been identified in Denmark with SARS-CoV-2 variants associated. Wow, with 12, including 12 with the unique variant. So they are doing genetic testing to tell the difference. That's good. Or they have I haven't seen any sequencing done on this. When it comes out, we're definitely going to be looking at it on the podcast, but because that's what's really interesting, where those mutations are.
Oh, yeah.
What type of mutations are they? And those mutations actually tell us something, right? They tell us something about the genome of the virus and what part is doing which thing, which is kind of cool. So Danish authorities have announced the following planned or ongoing public health actions. Good. Culling of all the mink. Good. Enhanced surveillance of the local population to detect all COVID-19 cases, including through thorough and population-wide mass PCR testing.
Good.
That's exactly what we were
Perfect.
talking about. That's great. Expanding the percentage of sequencing of human and mink SARS-CoV-2 infections. That's good. That's really good.
I'll sleep a little bit better at night tonight knowing that this is being done.
Yeah, rapid sharing of the full genome sequences of the mink variant, which is really, really good. Kind of like allowing us to learn things from the mutations. Yeah. I don't think that there's anything specifically wrong right now that we would be alerted by, that we would think that this would be incredibly dangerous. I mean, basically the same actions that we're taking right now in order to decrease the transmission of SARS-CoV-2 is going to help decrease the transmission of this mink variant too. So
Right.
cool.
Well.
All right.
Let's hope we can nip that in the bud before it becomes pandemic 2. Just
Yeah.
overlapping,
Exactly.
ongoing cyclical pandemics.
I know. Pretty incredible, huh? I mean, it's just one of those things where I think it really shows you the importance of doing things in the early stage, right? On the very early stage of all this happening, as opposed to after it gets out again and we have a second one that's in a global pandemic. I mean, kind of something that the scientific community has known for a long time, for example, to see how crazy a mutation is in SARS-CoV-2 and how rare this is. There's actually 2 variants of SARS-CoV-2 that we already know about and that we can kind of see through sequencing. It's kind of surprising to me that nobody talks about this in the media, but yeah, we actually have 2 strains of SARS-CoV-2 both in the pandemic right now, and that's actually through sequencing data we can actually see that those variants and the global spread of those variants.
Yeah.
So we can actually see where our populations in the United States came from based off of that sequencing data, because there's those unique mutations. We can kind of like plot it out. So we know, for example, that a majority of the SARS-CoV-2 within the United States actually came from Europe.
Right.
So when everything was being shut down and the United States administration was shutting down travel from China, for example, we actually know during that time that the travel restrictions were not on from Europe, and we actually know that a lot of people came in. They all heard that there was going to be a shutdown. They thought that there was, so they all traveled home, and we know through sequencing that a lot of our, probably, can't say a majority because we haven't had widespread sequencing of SARS-CoV-2 within the United States in order to determine that, but we know that a large percentage of the outbreak that was happening here was from the Europe infection.
Yeah,
So
from Europe. And you're talking about L type and S type, right?
Yeah, that was the paper in February that 2020 that came out and
Yeah.
talked about that. They actually hypothesized that there could be two different severities of symptoms too that are
Right.
associated with that. So that's actually a really interesting idea, because one of the things that people have talked about for so long right now is why are like SARS-CoV-2 positive people asymptomatic for so long, and why do so few people actually go into COVID-19? And then when they go, why if they do develop COVID-19, why is the severity so different? So there's like the severity of infectiousness that in that 2 to 14 day window, and then there's the severity of the symptomatic kind of, and they're both completely varying. So I mean, I just really wish we had done large scale sequencing in order to know that. It'd be
Right.
really interesting to see.
Oh, absolutely.
And that's important information too for like antibodies and like vaccines, like monoclonal antibody testing, like that, you know, that kind of stuff. The type of remedies. Once we know what's going on better, science can like kind of help guide that. All right. So we got some cool coming next. We got some really cool. The title is "How a Human Cousin Adapted to Changing Climate." A skull found in a South African cave suggests that the species
Whoa.
went through a process of microevolution during a chaotic environmental shift.
Look at that
Awesome.
thing.
Yeah, I mean, this is really interesting. I mean, if you go to an ecology class nowadays, I mean, you're actually studying how the environments and the uh, in, factors that that are spread throughout the world, how those differences change the genomes and change populations of animals and plants that live on them. So one of the ways of doing that is climate change. So it's really interesting to say what happened in the past and with different environmental shifts, and that happens a lot. It's called abiotic shifts. I mean, abiotic selection. So for example, like things that could cause that would be like any type of climate. So abiotic meaning not biological. So we're talking like the seas like flooding or something. We're talking about like islands coming together. We're talking about the islands being formed. We're talking about like separations of happening. So like let's say there's like a mountain range that goes between two populations, and now they can't mate with one another. So now they're
Right.
distinct populations. It actually happens a lot in nature. It's one of the
Right.
reasons why genetic diversity is so important, and why it's so key in order to keep diversity in the genetic population in order to deal with these environmental shifts. It is the earliest and best preserved specimen found so far of the species, which lives alongside and probably competed with Homo erectus. That is so cool.
That's really cool. And what did they say the name of the species was?
Paranthropus robustus.
Awesome. I
A
haven't heard of that.
large tooth, small brain, ancient human cousin.
This is a new one for me. I haven't heard of this
Yeah,
before.
I've never heard of that one. I mean, there's a lot of hominids that existed
Yeah.
on the world at one time. It's actually pretty crazy that there's still one. I mean, there's one right now. I mean,
Yeah.
kind of makes you wonder what happened during.
Yeah.
I mean, it's kind of
What did we do to the other ones?
exactly. I mean, it's probably a good. I mean, so and then also on top of that, like we actually have 3% Denisovan DNA and 3% Neanderthal DNA in us right now. So we can actually kind of know that there was some breeding and hybridization that happened during that time.
Fascinating.
All right, so I think I'm trying to.
2 million-year-old skull. Wow.
Okay, you know what? I'm trying to find. Oh, hello. Can you let him in? Yeah. Okay. All right, I'm trying to find like any type of thing that's in this. I hate I hate these science articles, because it's like they just bury the lead in the science. Like it's like three paragraphs of like, especially anthropologists, because they're like going around the world. They're like doing on these great adventures and stuff, and it's like, hello, Can we get to the science, please? You know, I think my breath stopped for a second. um, I mean, what is this? I think my breath stopped for a second. Is it really reading a novel? What is this? Um, I looked at her and I hadn't said anything, but she saw my face and she goes, "Yeah, that's what I thought." Oh my gosh. Miss Good would eventually learn that she had unearthed a 2 million-year-old skull that belonged to Paranthropus robustus, our large tooth, small brain ancient human cousin. It is the earliest, best preserved specimen around so far of the species, which lived alongside and mostly competed with Homo erectus. The skull provides the best known evidence of an ancestor of humanity evolving to adapt to a changing climate. Okay, around 2 million years ago, here we go. Here's the meat of it, finally. Around 2 million years ago, this area in South Africa is believed to have undergone a chaotic climate shift. The regional environment transformed from wetter and more lush conditions to drier and more arid ones. In order for a species like P. robustus to survive in such terrains, it would have needed to be able to chew on tough plants. But the specimen found in the cave at Jamolian didn't seem to fit what some scientists had previously stated about the human cousin.
Interesting.
Some scientists suggested that since they had found mostly large males in swatkrans and mostly small females in Dromolen, the size difference could be chalked up to sexual dimorphism, or the physical difference between males and females seen in species like manes and lions. That argument was that more or less only males lived in swatkins and only females in Dromolen. Okay. I mean, this writing, what is this? I hate this stuff. The day in the cave, she slid her finger beneath the dirt and felt a large sagittal crest on the top of the skull.
Oh my god.
Like, what kind of writer is this?
Yeah.
Like,
It's very
okay,
intimate for a science article.
Okay, we're just going to go to it. The abstract. Paranthropus robustus is a small-brained extinct hominin from South Africa characterized by a derived robust craniodental morphology. So something in the, large teeth. The most complete known skull of the species is DNH7 from Demolen Main Quarry, which differs from P. robustus specimens recovered elsewhere in ways attributed to sexual dimorphism. Here we describe a new fossil specimen from Demolen Main Quarry, dated from approximately 2 million years ago, that changes this view. DNH155 is a well-preserved adult male cranium that shares the DNH7, a suite of primitive and derived features unlike those seen in the adult P. robustus specimens from other chronologically younger deposits. This refutes existing hypotheses linking sexual dimorphism, ontogeny, and social behavior within this taxon, and clarifies hypotheses concerning hominin phylogeny.
Okay, so they're just saying that this is definitely a different species, right? From things that they've found before. That's all that said, right? Essentially.
No, it's saying that there isn't any.
Because before they were saying, okay, like it could just be like male skulls are bigger than females, so that's the sexual dimorphism.
Well, they're changing it. It was the same, but it's just changed. So we document small-scale morphological changes in P. robustus associated with ecological change within a short period of time and restricted geography. So what they're saying is that that's not completely different. It's just something within that it's the same species, but it's just different. It's just appearing different.
Cool.
And this is actually. This is a thing called in ecology. This is called phenotypic elasticity, which is actually something that's actually selected for within evolution. So you have a genotype. Your genotype is the genes. You think about your genes as kind of like the book that's writing the story, but then the phenotypic elasticity is like, oh, it's kind of like a choose your own adventure novel kind of thing. So you could actually have things part of your genome being turned on and off for different environmental reasons, and that's actually something that could be selected for. So within evolution, if let's say there's a population that's living in an environment where they constantly have changing environmental patterns, well, you're not going to want to constantly grow that really, really thick fur if you don't need it, right?
Gotcha.
So it actually is something that happens all the time. So for example, there's frogs and stuff that will actually only in mating times develop these spikes on their arms so that they can grab on when they're mating.
Oh,
so,
perfect.
Yeah.
Nice.
Yeah. So it actually it happens all the time. This represents the most highly resolved evidence yet of microevolutionary change within an early hominin species.
Okay, so I get it now. So they ruled out the other things like sexual dimorphism that could have been, and then that leaves environmental change over a small period of time. Okay. I get it.
So I think we stopped reading right when we needed to start reading. So in addition to the smaller than the P. robustus who lived at the Swartkrans, DNH155's cranium indicated its chewing muscles were not as strong as theirs. Mr. Martin said the differences suggest DNH155 and other P. robustus found in Dromaeosaurian were smaller, not because they were all female, but rather because they were earlier forms of the species belonging to a different population that hadn't yet been subjected to the environmental pressures.
Okay, interesting. I wonder what those environmental pressures were.
Environmental changes,
Yeah.
Some type of
was it hotter? I'm just
well,
interested.
they're thinking was the regional transformed from a wetter and more lush conditions to drier, more arid ones.
Okay, there you go. Interesting.
She named it after her dad, Ian. That's nice.
That's sweet.
It's also important that the study's authors didn't announce that the find was a new fossil hominin species. Instead, they asked the interesting question of how a known species changed during its evolution. Yeah, so I mean that's a really interesting question too. It's the species is kind of an interesting term that is as you get more into biology and really it kind of is an interesting way of categorizing things, because the kind of the high school definition of the species is two things that could mate and not make an offspring. So people will say oh, it's like a burrow or something, because you have a hybrid, and then those two things come together and you can't have a good you don't have a
viable
offspring. Yeah, which isn't necessarily true. That happens, but that doesn't mean that it's not a real species.
Gotcha.
For example, we were just talking about, humans are, we would call us Homo sapiens a species, right? I mean, we would say
Yes.
we were a species. Would we say Neanderthals were a species? Yes, we would say that. Okay, well, we know that Neanderthals mated with humans, and we have viable offspring.
Oh, okay. So
we're kind of just you listening to this, you are evidence that two species can mate and have viable offspring, and it's okay.
Gotcha.
So
we weren't different enough that we couldn't produce viable offspring.
yeah, exactly. And in that scenario is something, again, that is actually selected for within evolution. So, which is really interesting. You can actually have, for example, you can have things you can have there's different stop gaps and different things that could change and reasons why things wouldn't mate together. For example, their bits just don't fit together, for whatever reason. Let's say you see this a lot with cryptic species. Cryptic species are species that look exactly identical, but they they're mimics of one another. So they're trying to like steal the resources of another species and kind of like take advantage of that. And what they'll do is they'll have like the main species is like a triangle based hole, and they come out with like a square hole, and it's like their things like literally don't fit together, and they can't mate. They look exactly the same, and you'd think that they would be able to mate, but like there is actually an evolutionary reason why they didn't want to, because there was a push was to have two separate niches. They wanted to have their own thing, and especially parasites and stuff like that. Anyway,
the bit's got to fit.
Yeah, the bit's got to fit. All right, cool. All right, and our next piece of science news: artificial intelligence model detects asymptomatic COVID-19 infections through cell phone recorded coughs.
What?
How cool is that?
I'm gonna have to hear that again.
Results might provide a convenient screening tool for people who may not suspect that they are infected. So funny because they said asymptomatic infections, but then
they're coughing. So that's to me, that's not asymptomatic.
Exactly.
That's a symptom.
Exactly. Yes. Exactly. Asymptomatic people who are infected with COVID-19 exhibit, by definition, no discernible physical symptoms of the disease.
Right.
They are thus less likely to seek out testing for the virus and could unknowingly spread the infection to others. Not people here. Not on this podcast.
No, we wouldn't do that.
But it seems those who are asymptomatic may not be entirely free of changes wrought by the virus. MIT researchers have now found that people who are asymptomatic may differ from healthy individuals in the way that they cough. I mean, thinking about it, so these are SARS, I mean, again, we need to break out, it's easier to talk about the words. Yeah. SARS-CoV-2 is living in your body, and you have not exhibited COVID-19 yet.
Right.
But the SARS-CoV-2 has to be living within your body in order to therefore be living.
Right.
And you're not asymptomatic. So this is where the problem kind of arises.
Yeah.
Although there is something that's really interesting that, I mean, this is crazy. In order to delay the impact or to evade the host immunity, viruses will actually not express their virus genes yet. So they'll actually have, it's called a pathogenicity island.
Interesting.
And they have that's where all of their kind of like dangerous stuff is, but they won't actually express those genes until they come up to a population where they're like,
gotcha.
It's called quorum sensing.
Okay.
So they have this thing where they kind of like detect, like, is there enough of us? Is there enough of us? Okay, we were good. Boom!
Boom! Then
and
it
all
hits.
come on. They all turn it on at the same time, and that's kind of like what happens, and that's why you're infectious, and that's what's going on. So it's a really interesting conundrum.
Yeah.
So, what they're saying is that they could actually have an ability to detect those SARS-CoV-2 at smaller levels than you would think would be causing COVID-19. Go ahead.
Okay. So before they become symptomatic, because I'm always curious when they just say, okay, you're asymptomatic, does that mean you have SARS-CoV-2, but you've not yet begun to express COVID-19, but you will, and then you'll become symptomatic? Or is this somebody that now has COVID-19 and remains asymptomatic?
I think
curious.
COVID-19 is definitely is the disease that's like that's caused by SARS-CoV-2. So
right.
You could have
because you can have it for 14 days. You can have SARS-CoV-2 for 14 days and test positive and be asymptomatic and then begin to express symptoms later.
Yeah, exactly.
So I'm always like, maybe that's what they're saying in this paper, like before they develop symptoms, or are these people, they have COVID-19, symptoms would have begun to be expressed, and then still they just have this cough. That's all they have. So therefore they're asymptomatic. I don't know.
Yeah, exactly. It sounds like they aren't blowing through. Here we go. Here's the paper. Always good to go look at the paper. We hypothesized that COVID-19 subjects, especially including asymptomatics, could be accurately discriminated only from the forced cough cell phone recordings using artificial intelligence. What kind of models is it?
What?
There's so many different types of artificial intelligence. It's kind of like a buzzword. So I would wonder.
Yeah.
I'd wonder if it would be like deep learning, what kind of, oh, it's a neural network. Okay, sorry, I'm reading ahead. That's not good. Okay. To train our MIT open voice model. So that's the thing, is like, where is the artificial like intelligence learning? Like, where is that information coming from? That's like one of the biggest questions in artificial intelligence. Is it just like, is it training itself? Or is it, which is what happens with like Go and the chess stuff. You just give it the rules and it kind of like goes through all the iterations itself and trains it. Or you can like get a bunch of information and just like throw it at it, and then maybe that would then it could like, it's called deep learning, where it kind of like puts together things that you would normally see. So where does the information come from in order to train the artificial intelligence? To train our MIT open voice model, we built a data collection pipeline of COVID-19 cough recordings through our website and created the largest audio COVID-19 cough balanced data set
That's crazy.
reported to date with 5,300 subjects.
That's crazy.
Methods. We developed an AI speech processing framework that leverages acoustic biomarker feature extractors to pre-screen for COVID-19 from cough recordings and provide a personalized patient saliency map to longitudinally monitor patients in real time, non-invasively, and essentially zero variable cost. Yeah, because like, how do you know that the coughs are linked to COVID-19?
Right.
And that's one of the biggest things, is like, how do you know? It takes a lot of time to kind of constantly be testing people genetically. That's how I would do it, because I'm a molecular biologist.
Right.
So that I want to see qPCR data. I want to see that.
I know. This is so symptom based, and
Yeah.
not everybody exhibits symptoms the same way. It's so fascinating to me that there could be like a COVID-19 cough. I don't know what a COVID-19 cough sounds like.
Exactly. How would, I mean, they would have to have like a, yeah, I mean, I wonder, I mean, you'd have to have a backdrop, right? Like a control
Yeah.
group of like non-COVID-19 people, and you'd
Sure.
have to cough and have those coughs in there, but then
I mean,
compare it?
Yeah, I mean, and I guess I believe it because like whooping cough, for example,
Yeah.
that is like a classic cough. You know, you kind of like
Yeah.
bark like a seal. And so, you know, a pediatrician knows what that sounds like
Mm-hmm.
at that point. So like we've gotten good at listening to that. So I guess we're going to get good at listening to a COVID-19 cough too.
Yeah, exactly.
That's fascinating.
But these are asymptomatic people. So that's what's so crazy. Asymptomatic COVID-19 people.
I disagree
So they're
with that
SARS-CoV-2
wording.
positive, but then they have COVID-19. They don't have COVID-19. But I guess you could say that they are, because they have a cough.
That's
So-
a symptom.
Yeah, exactly.
How
what
is
you're
that not
saying
a
before.
symptom?
Exactly. So cough recordings are transformed with male frequency Kepstral coefficient and inputted into a convolutional neural network based
Wow.
in an architecture made up of one Poisson biomarker layer and 3 pre-trained ResNet 50s in parallel, outputting a binary pre-screen diagnostic.
That's fascinating.
So they did have like some type of, the Poisson biomarker, I believe, is the one that they're testing, and then 3 pre-trained in parallel. So they're comparing it with other ones. The neural network is an interesting idea. That that's like, the idea is like you have. Yeah, it's just like another. I don't know enough about it to talk about it. Maybe we could do a deep dive, like on our podcast.
I want to hear one of these coughs. Does the original article have
Oh yeah, that's a great idea.
like a link of the cough?
Yeah, let's see.
Because I bet there are people out there that have worked with COVID-19 since this outbreak began that could actually listen to the cough and be like, oh yeah, you've got it.
Oh, our CNN-based models have been trained on 4,200 subjects and tested on the remaining 1,000 subjects of our data set. Transfer learning was used to learn biomarker features on larger data sets previously successfully tested in our lab on Alzheimer's, which significantly improves this COVID-19 discrimination accuracy of our architecture. So that's what I was talking about before. Wow. So that's what they were talking about before. They're making it more accurate.
Yeah, I'm pretty blown away by that.
When validated with subjects diagnosed using an official test, the model achieves COVID-19 sensitivity of 98.5.
Wow.
For asymptomatic subject, it achieves its sensitivity of 100% with a sensitivity of 83.2%. Specificity of 83. What?
That's pretty crazy, especially because it's like over your cell phone too.
Like, how do you know? How do you know? Like, it's not just an artifact of the voice recording, and it is like
Exactly.
the cough. That's what's so
Because
crazy to
there's
me.
so many. Okay, so in nursing school, in order to
Wow.
like learn how to diagnose different types of heart murmurs and issues with the lungs, we would listen to these like robotic recordings, and they
Mm-hmm.
all just sound really tinny, and it's just like you're listening to a robot like try to make a sound. It's like you kind of had to. So there's really nothing quite like listening to it in person. So this is really interesting to me to that they're actually able to have such you know, like efficiency at diagnosing this, with like a robotic sounding recording.
Dude, this is really cool.
Yeah, that is really cool.
Dude, it's such like a.
I want
It's
to hear a
literally.
cough.
It's literally kind of like a meme in science right now, where it's like, oh, I have a problem, I don't know how to solve it. Just throw AI at it,
Yeah.
and
Let's have robots do it.
and this blows me away. I'm gonna have to read this on my own time because this is just really fascinating.
I'm gonna have to read this. Yeah, that's interesting. Okay, so did the article have a cough on there or no? I gotta know.
Let's see. It's not saying it. Wow, this is crazy.
That's a question that I have for nurses. Is there a nurse out there that's worked with COVID-19 patients that thinks that she could diagnose the cough with the same accuracy
good-
as
good
this
question.
AI? I would love to know that.
Oh, nice. They're making a user-friendly app.
Very
Okay,
good.
so they trained the model.
Oh, there we is that a recording? Down there
Uh,
with the icon?
no.
No. Okay.
This is them talking about it. It's like an interview or something.
I feel like I just heard it.
Let's see.
Yeah. Play that.
Oh yeah.
There it is.
Asymptomatic people who are infected with COVID-19 exhibit, by definition, no discernible physical symptoms between, for example, of AI when the AI model is COVID asymptomatic person.
Oh yeah. I'll tell you right now.
That sounds bad.
There's something in there.
Yeah. That doesn't sound good at all.
That you don't need.
Yeah.
I don't care.
I'll tell you
Yeah.
what. 6 feet back.
Yeah, dude. That's to have a freaking.
Yeah.
You're quarantined. You hear
Yeah.
that? You're quarantined.
Yeah, you go have that looked at, okay?
They found it was able to pick up patterns in the 4 biomarkers: vocal cord strength, sentiment, lung and respiratory performance, and muscular degradation that are specific to COVID-19. When the model is fed the cough of a COVID-positive individual who is exhibiting symptoms,
Ugh.
it is actually harder for artificial intelligence to discriminate. The researchers think it is because there are many conditions that create symptoms, such as the flu or asthma, and therefore the results are confounded. For this reason, they stress that their AI model is not meant to diagnose symptomatic people or determine whether their symptoms are due to COVID-19 or other conditions. The tool's strength lies in its ability to discern asymptomatic coughs from healthy ones. The team is now working on incorporating the model into a
Weird.
user-friendly.
So once you do have a bunch of symptoms, it can't just tell the difference between a bunch of the different types of infection, but it can tell the difference between a regular cough and an asymptomatic COVID cough.
That is fascinating.
Wow.
So it's like before you start getting sick. Like there's like that little window there.
Because it starts with something and then there's nothing.
Because a
They trained the
healthy
general-
cough, if somebody's just saying, okay, cough please, like
Yeah.
you shouldn't have anything in your voice. It should be.
Exactly, and here we go. They trained a general machine learning algorithm or neural network known as ResNet50 to discriminate sounds associated with different degrees of vocal cord strength. Studies have shown that the quality of the sound can be an indication of how weak or strong a person's vocal cords are. Sabrina trained the neural network on an audiobook data set with more than 1,000 words of speech to pick out the word them from words like the and then.
Wow. Oh my gosh.
All right.
Very specific.
Huh. Man, this AI stuff, dude.
Yeah.
I've got to figure out how to. I wanted to do some of it in my research, and I started going into it, and it's just like, woo! It is a deep, deep field, and it's like, it's actually. What's funny is that it's actually not getting to it, like using the software was not the hardest part. Like Google allows all their stuff to be free and open source, and you can get to it really easily. The problem is like knowing what to do. Like, why would you do one versus the other versus the other? There's so many different things that you can do. It's kind of like, you know, in a lot of science fields, it's like the Dunning-Kruger effect where you don't know what you don't know. So we just hear AI and we're just like, oh yeah, do the AI test. But then once you go into the AI field, it's like there's 50 different tests. It's the same thing with like, you know, I could tell you, oh, you just clone a gene. And then it's like, well, if you don't know anything about cloning, then you think, oh, you do the one thing. But in reality, there's 10 different ways of cloning a gene into a plasmid. And if you didn't know that, you wouldn't know that.
Oh my goodness.
This is crazy. This is the coolest thing
it's really cool, and I don't ever want to have that cough. That sounded bad. Especially after the chick got symptomatic later and they played her cough again. It was like, oh,
wow.
boy.
As they propose in the paper, pandemics could be a thing of the past if pre-screening tools are always on in the background and constantly improve. That is so true. I mean, seriously, I mean, this is such an interesting idea. I mean, they already use... You can use GPS, you know, to do it, and they did it in China in order to minimize the spread. They just had everyone have an app that said whether or not you're from an infected area and whether or not you could get on public transport. Which is, I mean, that would have helped so many. I mean, think about how many hundred, like, you know, you can't even propose a number because there might have not even been, you know, community spread if we did that.
Oh, we can't even get people to wear masks here. Like they're talking about, you know,
but that's just
transportation.
having a phone on your, you know, an app on your phone.
Yeah.
You basically have an app on your phone, and you're just kind of like you like cough into it or something. Or I guess maybe it says always on in the background. I mean, what would be the thing? I guess on Zoom.
Yeah, sure. Zoom's never going away. So
Zoom tells you like what your
it'll be Zoom.
Oh, James is gonna have to quarantine for a couple more weeks. He's got that COVID-19
yeah, just Zoom will let you know, like, oh, you could be positive. Yeah, stocks are going up. Zoom stock.
Oh, not right now because the COVID-19 vaccine. It's like all the
dropping down
COVID-19 stocks are going down. Yeah.
Oh boy.
All right.
I'm excited about that vaccine. Hopefully that's a real thing.
Yeah, I do too. We're gonna talk about that in the peer review.
Oh, good.
All right. So for our next piece of news, science news, I didn't mean to do this, but we got another way of using sound to discern it because of the sound episode of Catalyst.
Good.
So here we got, could listening to the deep sea help save it? In the abyss, everyone can hear you scream. All right. So basically, one of the problems that scientists are having right now, especially in ecology, and what we're trying to do is just catalog everything that's there. And that's one of the biggest problems is that, you know, people think, well, if something goes extinct, you know, you just bring it back. What's wrong with that? Well, if you don't even know it existed, how could you bring it back? And that's the biggest problem. It's not just like, you know, T-Rex. It's actually, you know, really, really small microbes that live in a community that are, you know, really hard to discern. And microbial communities, for example, like rely on one another to kind of like come together, and we don't know that they exist. So that one of the things that's happening right now is like a bunch of sequencing a ton of different organisms with the idea of that maybe we can actually preserve this even if we don't know how to bring them back, bring back a community of organisms, for example. At least we know that they were there, so that maybe in the future we could do that.
Yes.
And one thing, because of how well sound travels in water, so if you think about sound as kind of vibrations, and you have air molecules that are, hitting my mouth to the microphone, and then that's, transferring to the speakers. If those air molecules were, instead of having, those gaps being in between them, if they were connected, well, then those vibrations would just travel more easily from one molecule to the next, one particle to the next. And that's what happens in the ocean. And that's why sound can travel so far in the ocean. I mean, that's one of the reasons why you have, for example, like humpback and blue whales that are off the coast of Hawaii singing to one another off of the coast of California.
Wow.
So one way of that we're actually trying to preserve and catalog the, what's going on are soundscapes. So they're trying to record these places. I've heard of reefs are just insane. You listen to reef recordings and they're just constantly popping off. Like,
wow.
You get a lot of things that are happening. Yeah, I mean, sound is really important in the ocean, for example. Like famously, like whales actually use these things called SOFAR channels, which are if you imagine the, like I was talking about, how the ocean like comes together and water actually comes together, if those molecules are even closer together, well, it would travel even better, right? So and what makes molecules more likely to come together, closer? Well, you remove some heat. And heat's like pushing the molecules, kind of like away. But now the without the heat, they're like coming in with, together closer. So that's what the whales have kind of like, through evolution. The whales that use these cold, deep channels to communicate have, evolved over time, and that's what they do. They go down to these really great depths and actually use these SOFAR channels in order to communicate with one another.
That's so interesting.
Yeah, it's really cool, and which is also one of the friggin' reasons why. It's one of the reasons why fishing is so bad.
Right,
because
the boat noises.
Exactly, it pollutes those SOFAR channels. And I actually asked a marine biologist this once that was studying this exact SOFAR channels. Man, the conversations that you get in grad school are so fun, I'm telling you. One of them, and they were like, well, it's a good point, and they are completely polluted with engine noise, and that's completely true. But, they're going to use a different channel, so they could like dive deeper, they could use different calls, so yeah, it's really interesting. Anyway, so back to this. So they're actually, and I've heard of people doing this with reefs, and going with reefs trying to like see what the what was going on, but now it's really cool to actually see what these are. Oh man, I think we might be able to get these actual. Let's see if we can actually.
Yes, I want to hear a
it.
sound.
I know, me too.
I definitely don't know what a coral reef sounds like. That would be amazing.
It's so cool. Humpback whales. Oh man, this is really cool. Humpback whales they'll have it's like pop songs. They'll have like catchy tunes that will actually populate over time. You can actually, and I've seen it mapped, like the little chorus or something that they'll do, and then you can see it over time just take over the entire Pacific Ocean.
Oh my gosh.
Yeah, it's so cool.
And they really think the purpose of it is just entertainment, not communication?
Well, yeah, I mean, part of it, there's a bunch of hypotheses of what they're doing, but yeah, it could be mating, it could be alerting them of, for whatever reason. I mean,
they're all talking about it, whatever it is.
Let's just say this, like, however long humans have existed and have had science, we have constantly underestimated what
Yes.
the abilities of other animals are, the intelligence of other animals.
Oh,
so
I agree with that.
even if they aren't saying that they might be, like they probably are. I mean, we know, for example, that there's actual culture, like culture within whale populations.
Yes.
The culture is the definition of it within like the biology world is basically passing things down generationally. So
Yeah.
increasing knowledge and taking that knowledge and then transferring it generationally. So you have that with whales teaching one another where to eat, where to fish, what to do.
So this
Absolutely.
is the same theme. So again, it's a similar about 5, really more and more complicated. So this is a recording of, it's part of a song from the East Australian population from 2003. So what you heard there was a sort of moan and then a little whop at the end. So that was a very short, very simple theme that was part of the song in 2003. Then in 2004, the song evolved, so it had a few small changes to the song, but it's still it's the same type of song. So I'm going to play the same part of the song from East Australia, but from 2004. I'll probably do that again just because something popped up on my phone. Right
Yeah, I was gonna say.
I was like, wow, they're
So
getting
this
good there.
is East
someone's got a text
Australia from
notification?
2004, and this is the same part of the song
They got
that I
instruments.
played from 2003. So what you heard there was the same moan and the same little whop at the end, but there was a second moan in the middle. So now instead of 2 sounds, there's 3 sounds. So that's what we mean when we talk about grad changes to the song. These evolutions is they basically took the same theme of the song and they just added a sound. And in my research, that's what I was talking about when I said that the song got more complicated. So now instead of 2 different sounds, there's 3 different sounds. And now the whole thing is a little bit longer. Then in 2005, the song evolved again. So again, this is the same part of the song from East Australia, but in 2005. So now you have that same moan at the start, that same little whop at the end, and that little moan in the middle has now become a little moan and 2 squeaks, and they repeated that a few times. So again, it's a similar pattern, but now they've gone from 2 sounds in 2003 to 3 sounds in 2004 to about 8 sounds in 2005.
Interesting.
And so that's what we mean about them getting progressively more and more complicated. Then in 2006, the East Australian song sounded something like this.
Oh wow.
Okay.
So you have a moan and a bunch of really high squeaks,
Okay.
and
cool.
you can
Maybe get it. That's cool. I
That's
think
interesting.
the biggest thing, I mean, I think right now these soundscapes, as they're calling it, it's kind of cool. Like, but the biggest problem right now is figuring out what's making what sound, and that's one of the biggest things. Snooping on soundscapes, and they're talking about this in entire conferences that are talking about like what are making these like weird different sounds that they're hearing, what's contributing what, and it's hard to know without like, because you can't isolate them necessarily, right? It's not like something where you can just like, okay, now without the fish, all the fish be quiet.
Right.
So
It's the whole community that you're
yeah,
listening to.
exactly. So that's what they're talking about here. Like, how do we figure that it's a very new field? How do we figure this out? And I found some of the deep sea sounds. So this is the ambient sound of the deep sea. Oh yeah, by the way, this is a seismic air gun survey with echo sounders.
Yeah, I don't like that. That was very
Awful.
That was terrible. Wonderful.
Yeah, it sounds pretty much as yeah.
Yeah, it sounds like a gun
yeah, that's
going
right.
off underwater.
Here's the vent sound. Someone's doing
Okay.
some work there.
All right, much more subtle than the first one.
Yeah, I wonder if the ambient is with the thing that they actually had to subtract. Ooh, a whistle.
I could fall asleep to that.
Fish fins.
I think I've got some of these on my white noise machine.
All right, underwater communication. What?
That sounds like
That's so
like
cool.
a sound a human would make with like something digital.
Yeah, exactly, like an actual beep. That is so cool. Man, that is so cool. All right.
Interesting.
All right. Well, and I think, man, I was looking at like all these notes to all the scientists out there. Let's just like start uploading it to YouTube or something, people. I mean, like there's all these like conferences. Like it took me so long to try to find this stuff. I want to listen. You know, they're using SoundCloud, which is good.
Yeah.
I agree with, but all right. And let's see if you have one more. I think we might be going to peer review.
Okay.
All right. Yeah, I think so. I think we're at an hour, so I think it's a good time to go. All right. Well, Kristen, thanks for being on.
Thank you so
I
much.
Appreciate it.
I loved being here. Thank you.
Awesome. All right.
Okay, you get him in the peer review, Greg.
All right. Here we go. On to peer review. Here we go. All right. And today for peer review, we are talking about COVID-19, and we are talking about the Pfizer announcement and what that means for science, and what I basically want to talk about the response, how we responded to it, and what we could do better. So. So basically the only thing that's come out today that is this press release that says that there is a COVID-19 vaccine that has been tested by Pfizer and BioNTech, and that they had a phase 3 study, and it's 90% effective in preventing COVID-19 in participants without evidence of prior SARS-CoV-2 infection in the first interim efficacy analysis. Interesting. Okay, so this is people that haven't had it before, that didn't have the antibodies, which makes sense. So the analysis evaluated 94 confirmed cases of COVID-19 in trial participants, and the study enrolled 43,000 participants, which is really interesting to me. Like, for example, so with this, they're going to go and continue through to final analysis of the 164 confirmed cases in order to collect further data, and they are basically going to be trying to get the FDA to fast track this, which is good. The problem that I have with this is that people reacting to this as if it's a science peer-reviewed publication. And this is what we need to remember when we are doing these, when we have anything that's coming out. I mean, this is a press release made by Pfizer and BioNTech. So they are going to want to, and the CEO is out there talking about this, and they're talking about how great it is for the science and humanity. And that's great, and it could be. But the point is that we don't have a peer-reviewed study yet in order to say that. We haven't figured out what is actually going on here. Like, for example, my question is, like, how did they go from 43,000 participants to 94 confirmed cases of COVID-19? So it's kind of interesting. I mean, like, I'm curious about how they screened that. I'm curious about how things came out, and I think we need to be cautiously optimistic and can't rush to judgments. And I think that was that one of the problems that I saw is that, you know, people in the CDC, namely Dr. Fauci, came out saying that that this is great and this is a great milestone and all that kind of stuff, and I agree. But the problem is that we haven't seen the data, and we've seen other corporations like Johnson & Johnson, for example, pull out of their vaccine. So we need to figure out what exactly went on here and why the numbers are what they are. I mean, just having to their credit, they are saying that they are doing that. They are looking at that. They're going to put out the information, and but we need transparency. You know, we have to, you know, we have to do the science first. And I saw Dr. Fauci even said today that we don't want, you know, scientists in the new administration. He doesn't want them, you know, wants them to remain apolitical. But then at the same time, he was making this statement not based on science that this is an incredible, you know, day or, you know, incredible thing. And I thought that was just a little, you know, we need the CDC to be based off of science, and we need to be based off of peer-reviewed paper, not off of the press releases from the CEO. And that's what I'm talking about here. I'm talking about we need more science in what we're looking at and what we're figuring out, what this is you know, in order to go, you know, we need more science to figure out what's going on here. So I think that in general, this could be a good thing, and I'm not skeptical about the vaccine. If it comes out and it's gone through all of the phases, I definitely agree, you know, that people should be taking it, obviously, because it's gone through that rigor and it's been peer reviewed and the scientific community has looked at it. But I wouldn't rush to judgment on a press release that a CEO put out, because Pfizer obviously has a financial incentive in order to do this, like to say that this is as good as, you know, as good as it can be. So, and I think that's a fair characterization. I think that even Dr. Fauci, if he was listening to this, maybe he is. Dr. Fauci, thank you for being a subscriber. I appreciate it. I think that would be a fair characterization that even he would agree with. And I think that my larger point is that, like, the scientific community, like, is skeptical of one another, and that's fine. That's good, actually. Like, in order to be like a scientist, you're actually inherently skeptical, and that's a fine thing. We aren't all homogenous. We all don't think the same. All scientists don't go and move in lockstep with one another, and you know, science is based off of data. It's based off of transparency. It's based off of reproducibility. It's based off of it's based off of an inherent sense of skepticism, and I think that that's a fair characterization. And I think that I'm excited about looking through the paper, and we'll read the paper when it comes out about the vaccine when it does come out. But I think for right now, I'm cautiously optimistic, and I hope in the future we have a good publication that comes out that we can talk about. And I hope that this is the thing that that does, you know, that is a real vaccine. So, and in general, I don't think that telling people to, you know, be a scientist, specifically be more apolitical, I don't think that's a good move either, to be honest with you, because we need more scientists to be more engaged with things. We need science-based policy making. We need science-based practices, and we need more scientists to get involved, not less. So having people in prominent positions that are science-based, like the CDC, telling people to remain apolitical isn't a good look, especially right now with the reputation of the CDC being a little bit tarnished. Let's be honest. I mean, there's been a reason why there's been a levy and dozens of science-based publications that have never come out with a political recommendation ever saying that this administration specifically has to go, and because of the anti-science bias that's been involved there. So right now especially we need more people. Right now especially we need more scientists to get involved. We need more scientists to be reaching out to the public, to be talking about it, and to be saying when things are good and when things are a little suspect. And right now, I'm being honest with you. I want to see the review, the peer review information about that. But I'm excited to see when it comes out, and I'm excited to go from cautiously optimistic to optimistic. So in general, I think that science is better when we lift each other up. And by the way, I mean, when you're peer reviewing something, I mean, it is so brutal. So nobody is more brutal than a scientist to another scientist on a paper and a publication, because they are really going line by line, trying to make sure everything is okay. And that is one of the strengths of science. That is one of the things that really makes science stand out as an information gathering process, researching. You're searching for the information, and then you're having to research it because people might have shown a caveat that you didn't know. And honestly, I mean, to push back on my own view, a lot of these human trials are actually already put out there. So they'll actually put out the experimental information. So because humans are so expensive to work with, they'll actually put out their methods and their procedures, and people can actually comment now and push back and say, like, hey, are you sure you should be doing this? Like, isn't this a control that you want to look at? And that's really what it is. It's controls. So a lot of experimental design is mostly controls and how things are going. So I just wanted to say that I thought this was really cool news, and I can't wait to talk about it, but I think that it's a little bit premature to jump both feet in with a CEO press release. So thank you for watching. I really appreciate it. Thank you for Kristen for joining, and thank you. So if you liked what you heard, I really appreciate the like, the subscribe. Tell your friends, tell your science teacher, like, hey, this is kind of cool. We made this, the show is making science fun and approachable. And if you're a scientist, you want to come on, you want to talk about your research, I'd love to talk to you about it. If you have questions that you have about science, or you find a cool news article that you want to share and you want to have us talk about it, we have a Discord, we have a subreddit. So we want the engagement. I can't wait to talk to you and some of the questions that you have. So just let me know. All right. Until next time, thank you.